Hello and welcome back. This is Andre, and today we are discussing a classic method in genomics, and that is gene shaving, which is a method for identifying distinct sets of genes with similar expression patterns. Now, this paper is a great one, and it proposes a super cool method about clustering genes that have similar expression patterns. Now, there are, a million ways of clustering genes, but this one is particularly interesting because it manages to provide gene expressions that look like this. You see, so they have a clear structure in their expressions. Now, I'm going to explain exactly what this image is, but now that I've got your attention that this method gets cool results, let's move on. So, the problem is that they describe a statistical method, they call it gene shaving, that identifies subsets of genes with coherent expression patterns. That means the expression correlates highly, and large variation across conditions. That means across samples there is a high variance. They want to notice that gene shaving is different than hierarchical clustering and other clustering methods in the sense that genes may belong to more than one cluster. I'm going to explain how this is possible. And also the clustering can be supervised or unsupervised. Typically in clustering it's unsupervised, but now we also make it supervised. So now let's start with what are genes expressions. So these are genes expressions shown here when each row represents a gene and each column represents a sample. Without giving many details about it, I want to mention that so these genes expressions are nowadays a measure of how, so from genes you pass through a procedure, multiple steps, and there are proteins that are created into your body. So these genes expressions are measured by what proteins some cells in your body have created. But without many details, you want to notice that these values are normalized, and so these things are like positive expressions across a baseline, and the red ones are negative expression across a baseline. Imagine that the spectrum of the variation is, let's say, between 0, -1, and 1, and the green one is positive, above the baseline, and the red one is negative. Now, this is not the actual spectrum of values, but again, the key point is that they are more or less according to a baseline. Great. And these are genes expressions, and notice that some genes are so each row is one gene and it's expressed across multiple samples. Now notice that if one applies hierarchical clustering on both columns and rows, I'm not going to explain what's hierarchical clustering, but the point is there is a clear structure into the genes. Like, you notice some patterns. Now, the key idea is that ideally you want to have some clusters, meaning a bunch of genes which are correlated, meaning which are related to one each other, because there are multiple ways why would you want that. One way would be you want to reduce the dimensionality of this thing, like assume that each of those columns are a patient and you want to make predictions. Then sure that you can't make predictions with, like, in this case they have 5,000 genes, so 5,000 features. Other things are that you want to find biologically similar genes, possibly to do some later inspection. So finding related genes, it's useful for scientific discovery. And with these ideas there are multiple ways. Clustering is typically the most widely used, but in this paper, they proposed to find clusters. So each of these things are genes, and each of these things are, samples. And they proposed to find a method to find clusters where the genes are highly correlated. Notice that across those samples, meaning across these patients, this gene manifests essentially the same, very similar. But also notice that the variance, it's significant. This variance represents the variance of the mean. So you take those two things, you create the mean gene, you essentially you average them, and the average of that thing should be maximized. This is going to be part of the algorithm. This is another cluster which it finds, and this is another cluster. Now, surely, so these samples are sorted from the most intense one to the least intense one, but the point is that the method is going to find a cluster where the mean gene has very high variance. Now, why the mean genes, and how does this influence this shape? So the mean gene is going to be, averaged across every single column. So the mean gene should be, if you maximize the variance of that, you want, for example, this is one gene, let's say gene 1, and you have gene 2. Now gene 2 is in a good cluster. In order to have the max variance, it should follow like a similar pattern, because if you would have the G2 prime, something different, for example 3, 2, 1, meaning a different order, then the mean gene is going to be 4, 4, 4, and the variance here is basically zero variance, while between these two things the mean is going to be 2, 4, 6, and you have some variance, right? Because this is the mean and you have some variance. So by maximizing the variance of the average gene, this structure in the cluster is incentivized to appear, meaning the genes to express similarly across patients. That explains why each row is similar to another, and that explains also why the gene expression has high variance. Great. So these are a few clusters. Now notice that clusters can be of small size, can be of higher size, can be of smaller size. Now how do you define the size? They propose a method which is an automatic method. We are going to talk about it. Let's discuss the shaving algorithm. So we start with the entire expression matrix, each row center to have zero mean. So we have our expression matrix, where here we have the genes and here we have the samples. Now we compute the leading principal component of the rows of X. Now what does that mean? It means that you have, for example, this space. This is, let's say, sample 1 and this is sample 2, and you have some points. Regard that the points have been centered to have zero mean. So in the case of this space, they should be only on this sort of column to be really exact, but I'm gonna make an abstraction and I'm gonna assume that the points are, something like this, just for illustration purpose. Something like this, and maybe one here, and maybe a few one here. Great. Now we compute the principal component. Now the principal component is gonna be one line which passes through the center, and this line is selected such that if you make the variance of the projections across this axis, so if you make the variance of this point, of this point, of this points, it is maximized. So you compute this principal component 1. Now we shave off the proportion, so 10% of the genes having the smallest absolute inner product with the principal component. That means that we have our points and we want to remove some of them. We are going to do this iteratively. So at this point we are going to we get from 100% of the points, we remove 10%, so we get to 90. Then we remove another 10%, so we get to 81. We remove another 10% and we get to 72% almost. And we continue so on until we get to only one gene. So we are going to do this thing iteratively. Now, let's firstly talk about which ones were deleted, and then how do we do with this clusters, because at each step you have a different cluster. So here you have a cluster with meaning a set of essentially all the genes. At the other extreme you are going to have a set of one gene, but in between you have sets of a proportion, meaning some of the genes. Now we have computed a principal component and we want to select, the intuition is that you want to select the genes that contributed the least to this principal component. So remember that the principal component is computed, is essentially influenced the most by those points that have the highest, they contributed the most to the highest variance across this axis. So intuitively you want to remove those points that contributed the least to this variance. These points are going to be this, for example. You want to remove 10%, so maybe you will remove only these 2 points. So these points, clearly they contribute the least to the variance. They are very close to the mean. They just don't. So you remove those points, and now we got, for example, from the 100%, and now we have about 90%. So after we remove this 10% of the genes, and what I told you intuitively that you want to remove this which contributed the least to the variance, that can be computationally created as the absolute inner product with the leading principal component. Now, why with the absolute inner product? Let's maybe talk 10 seconds why. So if you have a thing, the principal component always has size 1. Now the inner product between one vector and a vector of size 1, it's going to be exactly, so the inner product between A and B, it's going to be this thing, meaning the projection of, A on B. So A dot B is literally equal with this bit. And this bit is essentially, like how far it is away from 0. In the case of the principal component, this is going to be, how much it is on this size. That's going to mean how much it contributed to the variance. Great. So we iteratively do this procedure to get with different clusters. And at the end, we are going to have a sequence of clusters. So each cluster we have in kg. Now, you need to estimate the optimal cluster size using a gap statistic. Now, the point is after this procedure, you want to pick one of these clusters of genes and say that, hey, all those genes are super highly, correlated. I want to make this cluster of genes to extract it, to keep it, because this is a good one. Now, they do this with the gap statistics, with glossing over, the formulas. This gap statistics computes a measure of what is the variance within one cluster of the mean gene. It is exactly what we have discussed here. Like it more or less computes the mean gene, and then it looks at the average, at the variance within the expressions of the mean gene. Now, this is the intuition, what it does. In reality, it computes, because all this paper is done on 48 patients. So in 48 patients, the risk is that there is very much noise, and the estimate of your variance is going to be wrong. So that gap variance essentially is an iterative procedure to estimate essentially how statistically correct is the variance estimate within a gene. So it's a fancy way to essentially pick the cluster with the highest variation within the mean gene. So now you pick one cluster, for example, this one. You keep this cluster and show it nicely into your paper to show that the paper works. But after you keep this one, you orthogonalize each row with respect to the mean. So you compute the mean gene in this cluster, and then you orthogonalize, essentially you orthogonalize the data. What does this mean? When you orthogonalize the data set, what you do, for example, you have, this is x1, and now we have this thing. Let's say that this is your average gene, and now you want to orthogonalize with respect to this. That means essentially orthogonalization is a process in which you want to essentially subtract this mean gene from this space. That means that you are going to project all the vectors onto delta x, and you are going, so you project them, and you are going to take the difference, meaning this perpendicular thing, the component of x1 that is perpendicular to mean x. So now the x1 orthogonalized, it's going to be a perpendicular vector of this mean gene. Now, why do you do this? You do this because you are going to apply this algorithm multiple times. You want to obtain a total of m clusters. At one total iteration, you compute only one cluster, but you need more. You want m. So you are going to iteratively run this algorithm multiple times. Now, each time you run these algorithms, you want to not get the same cluster. Like, you want to have essentially a different cluster. You want to ameliorate the influence that the cluster that you have already found has on the data, on the next clusters. And one common way to doing that is to orthogonalize, because when you orthogonalize, we are getting into details now, but when you orthogonalize with respect to a vector, what is going to happen is that this vector is going to be essentially zero, because it has no component in it that is perpendicular to it. While the x1 prime is like this, the mean is like this. An interesting thing of this method is that it allows for a gene to appear in multiple clusters, because you orthogonalize with respect to the cluster mean. This means that, for example, if this is the mean, for example, the cluster points could be, around here. So when you orthogonalize, the cluster of points is going to move around, call it around 0. Now, the next time you are going to find a principal component, assume that you, after orthogonalization, you also have some other points. Clearly, in this case, the principal components want to capture the most variance. It captured that. But then the ones with the points with meaning the genes with the largest inner product with the principal component are going to be, on the edges. So those points are highly, less likely to be selected in a new cluster. Now, highly unlikely does not mean impossible. So a gene that was selected in a cluster, then orthogonalized, it can be again selected for a new cluster. It's not impossible, but every time you orthogonalize, you essentially make that gene closer and closer and closer to zero. Now it's gonna come a time when you made essentially most genes close to zero, and that is going to be the time where they're going to be selected again. But the key point I want to emphasize is that a gene can be selected multiple times in a cluster. And the orthogonalization process essentially shifts this around zero. And this is gene shaving. This is literally the method. It gets this great clusters. Now, this method is fully unsupervised, but they also propose an extension for making it supervised. Now, before that, I want to show some of the results that they've got into the paper. So let's look at this figure, figure 7. This is the percentage of gene variance that is explained by the first J gene shaving column averages. What does that mean? So this is the PCA explained variance. It's essentially how much variance is explained by a number of principal components. So you see that, for example, when you have 10 components, about 60% of the variance is explained. That means the variance of the projected data in the PCA space has 60% of the variance of the whole initial data. Don't get lost. But here we see, so this is the first cluster of 8. Now, in the next cluster, you found 25 more genes, so in total you have 31 genes. Now, in the next cluster, you found another 2 genes, so you have 33 genes. Now, these 3 clusters of 8, 25, and 2 are exactly the ones that I've been showing here. These clusters. So each gene represents a vector in a space, in the space of samples. Now, if you project the data onto these genes, the variance in that space is essentially a percentage. Now, the key point of this figure is showing that on the x-axis you have the number of clusters. 2 clusters, 4 clusters, 6 clusters. And the axes of this space are the mean, let's say, of cluster 1, the mean of cluster 2. Obviously, those are not going to be perpendicular like in PCA, because PCA finds an orthogonal space, but they are going to be, let's say, a relatively good axis for a new space, in the way that it still preserves a relatively good variance. The point here is to show that the clusters represented by their means are somehow more or less perpendicular, meaning they are part of, if you look into the space, it's going to be maybe a cluster here, maybe a cluster here. The average is, let's say, relatively perpendicular, but not so much. And this is great, because it shows that this method is capable of extracting, clusters from different parts of the space, which is good. Great. So we have supervised shaving. Now, previously we saw only unsupervised method. Now, in the supervised gene shaving, the key point is that you want to pick the cluster size supervised. Like, you want to inject some information that would help you pick the cluster size. In the case of the previous, this thing was computed using the statistic that essentially maximized the variance across the mean gene. It did that more or less automatically. Now we want to pick the cluster which specifically maximizes the variance across the mean gene. It's essentially the same principle. We want to obtain the same thing, a mean gene with high variance, but just that in this, call it optimization function, you also include a supervised term. And this term is, this thing can be labels, these things can be, for example, patient survival data, meaning times, like regression or labels for classification, and you compute the thing. Now, to me, it's not clear because this is a vector and this is, a class, it's another vector of labels. It's not super clear how this J function is computed, but imagine it as being just a loss of how, call it predictive are your clusters from for the class label. And of course you take this and you add a hyperparameter. I don't like hyperparameters, but many times not necessary. The question is when would you want to use this? And the answer is when you want to find clusters of relevant genes with respect to a specific task. For example, you do patient survival, like how many months the patient's gonna live, or you classify with respect to a specific type of cancer, breast cancer. Now, you want to find genes that are important for that cluster, for that task. You want the genes that are important for breast cancer. So in this way, you are finding clusters that are specifically tuned to be relevant for predicting your task. This means that you apply supervision for. This supervision again comes only when you pick the number of genes in a cluster, but in this case, it picked a cluster of 234 genes. It's quite a few genes. Notice that it preserves some of the pattern that we saw in the first thing. Like in this thing, I think it was selected with like to be a relevant cluster for patient survival. But the point is that it still preserves some of the properties, meaning the genes are relatively correlated and they have a high variance of the mean thing. The point is that these genes are relevant for our task. And this is a supervised way of obtaining things. Now, this is everything that I had to say. Overall, it's an interesting paper. It's clearly very cool, I would say, the idea of shaving multiple times, and it's cool that it gives some great clusters. It's also another takeaway from this paper is how they manage to essentially embed like the idea of having clusters with a high variance mean gene. Many takeaways from this paper. Now, some cons about this paper is that the method is a bit complicated to implement, and this is never good for a paper. But this one seems to me like super complicated to implement. I looked online; there are essentially no implementations on GitHub, so that's a con. But there are takeaways in terms of methodology which are interesting. Now, if you enjoyed this video, click that subscribe button, and I hope to see you in the next one.